My name is Qian Zhu. Today I'm presenting my poster CUT&RUN Tools, a flexible pipeline for CUT&RUN processing and footprint analysis. So CUT&RUN is a protein DNA mapping technology. It stands for cleavage under targets release using nuclease. And this technology was developed in the lab of Hanikov in 2017. It involves the use of a special fusion protein called protein A fused with a micrococcal nuclease, and this fusion protein gives it the ability to recognize a TF-specific antibody and cuts around the TF to generate fragments that can be sequenced and analyzed further for binding sites. And in comparison with previous ChIP-seq, this technique does not involve the use of cross-linking, and it also generates a higher resolution mapping due to the fine cutting of the MNase enzyme. And other advantages can be summarized in this table, showing also that it requires fewer cells, fewer reads to be sequenced, while maintaining high signal-to-noise ratio. However, CUT&RUN does present a few challenges. First, it is a paired-end sequencing technique, which requires a high percentage of pairing of reads. Second of all, it produces short fragments due to the fine cutting of the MNase enzyme. The fragment length ranges from 50 to 100 base pair, and it is frequent that the fragments actually are shorter than the read length, presenting the possibility that some chunks of the read might be contaminated with adapter sequences. And lastly, CUT&RUN offers the unique opportunity to look at single locus cut frequencies at individual base pair, and this usually was not taken advantage in previous peak-based analysis. And the useful things of this cut frequency is that usually the TF binding locations are situated within the regions of no cut, such as indicated here. And this may not be immediately obvious from the fragment pile-up. To solve these challenges, we have developed a pipeline called CUT&RUN Tools, and this schematic shows the three main steps. First, the CUT&RUN experiment generates a paired-end R1 and R2 FASTQ files, which are aligned with the typical aligner Bowtie2 to generate BAM files and visualize in the bigwig format. Then, the BAM files are subject to typical peak calling using MAX2 or the seq-R analysis tools to generate a set of peaks, and then we do additional de novo motif analysis to find a set of motif candidates that might represent the recognition sequence of the antibody. Then we use these motif regions to estimate the single base pair cut frequency to generate a cut matrix that can be used for estimating single locus log odds of binding, as well as plotting a motif footprinting pattern as shown here. Perhaps the underappreciated aspect of CUT&RUN is this cut matrix estimation of binding log odds. And here I use the GATA1 as an example. Here we have performed CUT&RUN in CD34 expanded cells. Here first we show the comparison of CUT&RUN and ChIP-seq, showing the higher resolution of CUT&RUN peaks in terms of narrower peak width. On these peaks, we further performed de novo analysis and found the HGATA-A to be the highest enriched motif among CUT&RUN peaks. And furthermore, by plotting the cut matrix around this motif and the surrounding region, we immediately see a high accumulation of cuts on both sides of the motif, while seeing no cuts in the motif center. And the protections here shows the protections of the DNA due to TF binding around exactly the HGATA-A motif here. and immediately surrounding region have high accumulation of cuts. And this pattern is the stereotypical footprint patterns of any given TF. And it could be very informative to reveal how the TF binds to the DNA. And what is more, we go one step further by utilizing the motif footprint pattern to help us estimate the single locus binding log odds. And what this is as shown in this formula, which is the probability of the overall cuts in the region PR given Z, as well as the conditional probability of the spatial distribution of cuts given the total of cuts in that region. So in summary, we have presented this pipeline, and this pipeline allows people to estimate footprint as well as the single locus binding probability. One specific question that we usually get was why do we need the log odds analysis when we can just simply estimate binding sites from doing a peak calling analysis? And the answer here is that actually log odds can sometimes disambiguate very closely located motif to find the likely binding sites. And in this case, we are using the NFYA cut-and-run as an example. This region of HBG1 and 2 has two twin CCAAT motif of NFY, and it is not immediately clear from the pile-up picture which one is the more likely binding site. However, by looking at the single locus cut frequency, we immediately see that the estimated log odds are very different. One is highly positive due to the resemblance of this to the stereotypical footprint. The other one has highly negative. So in this case, NFY footprint indicates a protection of 7 base pair and 13 base pair, which is present in the proximal this region here, but not in the distal, which have also done additional functional assay and CRISPR motif editing to verify that the proximal is the functional motif. And I thank you all for listening and for your attention.